Hello, hi everyone, and welcome to this lecture on active colloids at fluid interfaces for this ITN school on experimental methods on active matter. So I'm very happy to be able to give a lecture to you today. My name is Lucio Isa. I'm a professor at ETH in the Department of Materials, and obviously we work on active colloids. So in particular, the topic of today's lecture is going to be active colloids at fluid interfaces, and throughout the lecture, you sort of see why these systems are important, and we go through some important aspects of their characteristics and their behavior. So in this school, you've already looked at many different cases in which we have looked at sort of artificial microswimmers or active colloids in bulk situations. And very often when we talk about these systems, we sort of think that year zero for experiment in these type of systems, especially when we talk about catalytic Janus microswimmers, starts in 2007 with a beautiful experiment by Housey and et al., which basically looked at the self-propulsion of Janus particles half-coated by platinum in the presence of hydrogen peroxide, and how the selective chemical decomposition of the hydrogen peroxide gave rise to self-propulsion. But perhaps not many people know that, in fact, at least the first example of a catalytic swimmer that I know about was not this one. It was not carried out in experiments where the microswimmers were in bulk, but was instead looking at the confinement of such catalytic swimmers at an interface, and in particular a fluid interface, and in particular at an air-water interface. So this work that predates sort of these experiments by about 5 years comes from the group of George Whitesides, and here they were interested in looking at the dynamic self-assembly of self-propelled object freely floating and moving at an air-water interface. And in this case, the swimmers that they made were larger, significantly larger in size, but essentially consisted of a PDMS, so silicon elastomer plates, that had a porous platinum-covered, let's say, engine that was inserted into an aqueous solution of hydrogen peroxide. These objects were floating at an air-water interface, and again, the catalytic decomposition of the hydrogen peroxide by the platinum was responsible for the chemical gradients that gave rise to propulsion. And here they were in particular not so much interested in the dynamics and in the propulsion of the systems as active particles as we think about them now, but they were mostly interested in the possibility of having dynamic assemblies of such objects floating at an interface due to capillary actions. And by changing the wettability of these objects, they were able to introduce both let's say, attractive and repulsive capillary interactions, and looked at the dynamical assembly of the self-propelling interacting objects with one another. So you see that even though the vast majority of the work on active particles has focused on particles in bulk, indeed, interfaces were present in this field right from the beginning. And fluid interfaces in a very general way are actually present in a very broad range of different materials and situations. And in particular, we can identify the case where we have an interface between a gas and a liquid. And this is typically found in the case of foams, where we have gas bubbles that are surrounded by thin films of water typically. And so we have an air-water interface, which is the main constituent of a foam as a material. Another very common case is not between air, water, but is between two immiscible fluids, typically oil and water. And oil and water interfaces are dominating the behavior of materials such as emulsions, which are droplets of a fluid phase dispersed in another immiscible fluid phase. And they can either be water droplets dispersed in oil or oil droplets dispersed in water. And essentially here we have an interface between oil and water, which is again the main constituent of emulsions as a material. And also fluid interfaces are very commonly found also in biology, in biological materials. In fact, we can think about the lipid membrane that is enveloping cells, for instance, as a fluid interface where we have a hydrophobic compartment and a hydrophilic compartment, which are separated by the interface, by the membrane itself. So due to this reason, I mean, there's been a huge, let's say, interest in understanding the behavior of fluid interfaces for materials development, as well as for fundamental understanding also of biological and biologically inspired systems. However, fluid interfaces have also emerged as a perfect template of where we can construct and assemble two-dimensional materials constituted by solid particles. By this, I mean that the interface itself acts as a place where we can absorb and investigate solid objects like colloidal particles. This is coming from three main ingredients, which are also very much related to what I'm going to tell you today. And the first one is that if we take a solid object, say a microparticle or a nanoparticle, and we place it at an interface between two fluids, say air, water, or oil, water, the presence of the interface is able to confine the particle within the interface plane, essentially in an irreversible fashion. So we will see that for particles that are larger than a few nanometers, the trapping energy that keeps a particle at the interface greatly exceeds thermal energy in such a way that this binding, this vertical trapping is irreversible. So this means that if we can place the particle at an interface, basically essentially for free, we had a two-dimensional or quasi-two-dimensional material where the particles follow the shape of the interface. But importantly, even though the particles are trapped vertically across the interface, they retain lateral mobility. The interface itself is fluid. The particles can move around within the interface, either by Brownian motion, but as we will see later in this lecture, also by active Brownian motion, so that the particles can move around. And also very importantly, and we're not going to talk too much about this in today's lecture, but particles at interfaces also display some specific interactions which are absent or very different in bulk, which make it possible to assemble materials and to have particles interacting in ways in which would not be directly attainable in bulk. So even though the particles at fluid interfaces have been studied in many different cases and at many different viewpoints, the work when one looks at active particles at a fluid interface, in spite of the origin of the seminal contribution that the study of such systems have given to the development of catalytic microswimmers, for instance, remains very scarce. And indeed today I'm going to tell you something about our own research as well as some general research in this topic. So when we think about colloidal particles confined at an interface, the motivation behind looking at active colloids at an interface relies on different aspects, which are pretty much the ones that I was mentioning before. So as I said before, particles at an interface are irreversibly trapped, and that also holds true for active particles that we will see in a moment. Particles can move within the interface, and that also holds true for swimming at an interface, as I will give you examples. And in addition to the development of new interactions or different interactions within the interface, the presence of the interface also enables different means of propulsion. And in particular, as I will show you towards the end of this lecture, very efficient means of propulsion. So in that respect, I really believe that examining and studying active colloidal materials in two dimensions at a fluid interface offers significant interest and promises for the realization of two-dimensional systems. So today's lecture actually will focus on different aspects and different topics. The first part of the lecture will be dealing with studying how particles absorb at an interface and how they're trapped at an interface, and why the trapping that we've mentioned already a few times is actually irreversible, and how that affects, for instance, the case of a Janus colloid at an interface. Then we will go through some details of the measurement in which the position of a particle at an interface, which is described by a quantity called the contact angle, can be measured. And then we'll tell you something about different ways in which the surface tension or the interfacial tension between the fluids can be measured, because these quantities are very much important in the description of the behavior of active colloids at an interface. We'll then focus on two examples that are coming from recent work in our lab, where we first focused on catalytic Janus microswimmers at a fluid interface, and we will see how all of these quantities are important in describing the behavior of active particles in an interface, and finish by discussing a propulsion mechanism that is exclusive to fluid interfaces, which are Marangoni stresses.